Introduction to the Mouse Genome Informatics database. MGI is the comprehensive international database for the genetic, genomic, expression, and phenotypic data in the laboratory mouse. MGI was initiated along with several other model organism databases as part of the Human Genome Project in 1989 and has been supported by the NIH for 25 years. The data in MGI come from a variety of sources as depicted here. We have a staff of scientific curators who constantly comb through and read peer-reviewed publications. We accept direct submissions by researchers, and we have a number of large data loads from other projects and sources which also feed in. Data is updated to the public site weekly. To access this data, users can begin at the home page. There is, of course, the quick search field at the top, which accepts text searches for genome features, such as gene name or symbol, or vocabulary terms such as a disease, a phenotype, a function, or protein domain. In this box, MGI allows multiple entries separated by spaces or commas, quotes for precision on longer phrases, and use of an asterisk as a wildcard. If you look below, you can see some of the types of data in MGI and access more information for these categories and topic-specific search or analysis tools by heading over to the appropriate MIDI home. For some of our longer-term users, you may notice that we've recently added some sections here. The human-mouse disease connection, a more straightforward link to access batch data files, along with some new analysis tools for those, a series of guides to standard mouse strain, gene, and allele nomenclature at the bottom. If you're looking to run a search that allows you to specify parameters beyond a simple text match, you can also save yourself a click by selecting a query form from the search menu at the top of the page. But rather than go there, let's take a brief look at what else is on the MGI homepage. You'll see here there's a number of informational and tutorial documents on mouse genetics and how to use MGI. Over to the right, there's an image carousel where we highlight new tools, indicate meeting attendance or upcoming workshops, and make other announcements. A static announcement section is below if you want to see what else is new, browse some of the older announcements, or read more detail about a release. And you can keep in touch with us via social media or send an email to ask for any information or if you have any questions. To move on in this quick tour, we'll start with a quick search for the gene apolyprotein E using the gene symbol. The quick search will return a list of matches ranked by best match and separated into genome features and vocabulary terms sections. You can see here that our exact match, the protein coding ApoE, is the top ranked hit, and the far right column gives you an idea of why this particular feature was mapped, which can include synonyms, orthologs, or other species, orthologs in other species, or partial matching, including a gene name. Below the genome features is the vocabulary terms, and here you can see that returns things like functions, diseases, protein domains, and phenotypes that all have some relevance to an ApoE search. You can click on the associated data to see the genotypes, mouse models, and other annotations. If we go to click on the protein coding gene symbol for ApoE, this will bring us to a gene detail page. At the top of the gene detail page, you'll find the ID and synonyms. Right below, position, both in coordinates, positional coordinates and linkage, vertebrate homology information, diseases and alleles, function using the gene ontology, so that's actually biological processes, cell components, and function. Gene expression, which is hosted by the Gene Expression Database as part of MGI. Sequences and polymorphisms. Protein links. And then references for all the data in MGI, all the publications that refer back to ApoE. So if we zoom in on the diseases and alleles, you'll see that I've expanded out both the human homologs and the alleles and phenotypes section, which refers to the mouse. And you see here where I've boxed the disease associations. ApoE is involved in 6 different human diseases. You can click on that to generate a pop-up and see which those are. And this data is provided to us by OMIM. On the mouse section, you can see that ApoE mutants have been used to model at least 2 diseases in mice. And click on that, you can see that these 2 diseases as well, the apolyprotein E and macular degeneration. What are those mutations and alleles? Here you can see that there's a list of all the alleles that have been described and are included in MGI. And to define these a little bit in MGI, using generation type, we'll start with the gene-trapped alleles, where a gene-trapped allele is a random cassette insertion. The resulting allele may be null, hypomorphic, or functional, and they're typically reporters. Another type would be the induced or spontaneous mutation types. These can include chemical induction or radiation induced, or just random mutagenesis. These are also random localization, and the resulting allele may be null, hypomorphic, functional, or gain of function even, or include missense, nonsense, and rearrangements. A targeted allele is actually the first one where you have a homologous recombination in a specific location within the genome. Here the vector introduces a cassette that replaces exon 2, and the resulting protein does not contain exon 2 and is therefore non-functional. As an alternative, another type of allele that is targeted is a floxed allele, where you introduce recombinase recognition sequences indicated by the pink triangles. These recombinase recognition sequences are by themselves non-functional, producing not a functional component. So the resulting allele is perfectly normal until a Cre recombinase is co-expressed, in which case the floxed or flanked region will be recombined in some way. So targeted alleles, of course, are targeting a specific locus. They may include a specific introduced variant. They may be null or knockout. They may be missense and knock-in, or they may be conditional floxed. A transgenic allele is the introduction of a foreign DNA sequence for expression. This may be non-mouse DNA. It may disrupt an endogenous gene or it may not, and this includes recombinases such as Cre and Flip, which come from a bacteriophage or yeast respectively. The last kind of allele type I'm going to describe is the incidental mutations, which is a new one for us. And these are introduced germline variants from ENU mutagenesis that are at a distance from the variant described phenotypically, and are actually of unknown significance, so they're not characterized or not thought to contribute to the studied phenotype. They are, however, reported to us. So if we return back to the alleles and phenotypes section on a gene detail page, and click on that 40 for all alleles, you'll see a phenotypic allele summary. Here you've got the first column with the allele symbol. You'll notice the allele symbol takes the gene symbol and adds a superscript. For spontaneous mutations or historical mutations, often that will just be an allele ID. In the case of targeted mutations, they take the format of superscript TM1 or TM followed by a serial number, and then a lab code such as ApoE TM1 BRCS. The category is another column that's particularly important. You can sort that using the arrow keys. And you can see here that the different categories of alleles, including spontaneous, transgenic, targeted. And if you click on one of those, you'll go to an allele detail page, which describes it in a lot more detail. This is the ApoE SHL allele detail page. You can see there's a mutation description right there. A small triangle, if you click it, it exposes the mutation details, as well as a J number that links you back to the original publication describing this. If you're looking to obtain mice, the Find Mice row is where you want to look. You can see that there's a link for one strain that has been deposited for commercial or public distribution. And then there's a list of references that have actually used this particular allele. In the center of the page, you'll also notice a phenotypes and genotypes table. That in these, you have the phenotypes observed, which are annotated to specific genotypes. So HM1, which is composed of a homozygous ApoE SHL on the genetic background just to the left there, the B6 core background. HM2 has the same allele composition but on a different genetic background. And each of these rows correspond to a column in the phenotypes table. Occasionally background effects will make a difference, so you may notice that some of these with the exact same allele composition have slightly different phenotypes described. You can expand these into more specific structured vocabulary terms by clicking on the triangles to expose the details, and then clicking on the check marks or column headers to bring up a free text description of what each of these observations actually is. Each observation will also be tagged with a J number that will return you to the publication that initially described it. So in summary, MGI provides data and tools for the research community in a relational database. Genes, alleles, and phenotypes are described using searchable structured terms as well as more detailed free text. And each piece of information is cited with a J number, which will refer you back to the source of the information. If you have any additional questions, please feel free to contact mgihelp@jax.org, and we'd love to hear from you.